With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello students, have a look at the question. Question is function of ORI site in a vector is to. Options are initiate insertional inactivation, initiate replication, codes for proteins involved in replication of plasmid, initiate antibiotic resistance. So first of all, if we'll see a vector which is a vehicle molecule used to carry the foreign DNA, and the major characteristics of a vector is it should have ORI. What is ORI? We will see it just later. And secondly, it should have certain selectable marker. That selectable marker could be antibiotic resistance, blue-white screening. So it could be anything, and it should have selectable markers. Now we will see what is ORI. ORI, it is origin of replication. It is the full form of ORI is it is a origin of replication. Origin of replication, it is a genetic sequence, it is a gene sequence present in a vector. We can say that the origin of replication, it is a gene sequence which are present in a vector. This vector could be plasmid, that could be any bacterial artificial chromosomes, it could be any vector. It is a gene sequence present in a vector, and the main function of this is it can from it the replication can start. Replication starts at this point, and this is the most characteristic feature for a molecule, for a DNA molecule to be called as a vector. It should have the ORI, means it should can replicate independently, and that replication start at this point. Now what is the purpose of having the ORI? As we all know, vector, it is carrying the foreign DNA. So if vector carrying the foreign DNA and it is having the ORI, it means it will replicate, and it will also make the multiple copies of that DNA. It make multiple copies of DNA along with its replication. Multiple copies. Then when the multiple copies of the transgene will be made, it will be very easy to then transform into the host cell. So because if it would be a single copy, then there is very less probability to enter. If there are multiple copies of transgene along with the plasmid, then it is very probability is very high to enter the plasmid along with transgene into the host cell. That's why the ORI, or origin of replication, is must in a vector. Now, function of ORI site in a vector is to initiate insertional inactivation. This is wrong. That is a function of the that is one of the characteristic of the selectable marker. Initiate replication. That is very correct. Codes for protein involved in replication of the plasmid. This is wrong. Initiate antibiotic resistance. That is again the characteristic feature of the selectable marker. So this is incorrect. Correct is option B. Hope you get it. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.